Good morning. Today I have with me Laura Vella from the Florey Institute. Welcome, Laura.
Hey Jodette, nice to be here.
Lovely to have you here. Laura, I might let you do introductions on your research topic because I can't pronounce it.
So Jodette, so I'm from the Florey Institute for Neuroscience and Mental Health and also the University of Melbourne. So I run a research group and we predominantly look at what are called extracellular vesicles, but really they're bubbles. They're little bubbles that each cell in our body actually release, and they release them into our bodily fluids. So we can find these little bubbles in our urine, in our blood, in the fluid that surrounds our brain, any fluid that you can think of. We've found these little bubbles in, and these little bubbles, the reason we're interested in them is because they contain a lot of information from the cell that they've actually come from, or the tissue. So for example, the brain and the cells in the brain release these little bubbles, and they do that constantly. So whenever there's some issue with the brain, these bubbles, the content of them will change to really reflect what's going on in that particular tissue.
Human body is so complex. I was just saying. Yeah, keep going, please.
So we analyse these little bubbles. So we are interested to know the function of these little bubbles, but also to know what they can tell us about the brain in various diseases, and particularly in Parkinson's disease.
Right, great. So we do a standard interview. People who are watching would have seen a couple of interviews with people from the Florey by now. And we always start with the standard question. Tell us a bit about yourself, Laura.
So I did my PhD at Melbourne University a few years ago now. So I finished in 2008, and I was working on a neurodegenerative disease. So neurodegenerative diseases include Parkinson's disease, but also Alzheimer's and another type of disease that's called prion disease. And that's what my PhD was in. And it was there that I really started to get interested in a lot of these different diseases, even though they all come under one umbrella of being a neurodegenerative disease. And so I was spending a few years studying prion disease, and then I had a look for a couple of years at Alzheimer's disease. And more recently, been having conversations with colleagues about Parkinson's disease, and realised some of the similarities and overlaps with some of the work I'd done previously. And so that had really, I suppose, sparked my interest in this particular disease. And the last year, we've now started up 2 projects in the Parkinson's disease research area.
Okay, well, let's have a quick chat about those projects because they sound very interesting.
Yeah, so they all relate to extracellular vesicles, and trying to understand what those vesicles can tell us about, I suppose, really early Parkinson's disease. And something that I wasn't aware of, and a colleague over a coffee actually sort of said to me, do you realise that when Parkinson's disease is actually diagnosed, it's when patients have these motor impairments, but quite often, in most cases, patients are having some other symptoms very, very early on. And I said, well, how early on? And my colleague said sort of 10 to 20 years. And that was really surprising to me. And I thought, well, and he said, what we need to be doing is actually studying people at this really early phase. And it's then that we could potentially diagnose, we could potentially treat. That conversation was probably a year ago now. And I realised that we could start having a look at some of the early processes, I suppose, that happen in the disease that happen really early on. So we've got some cell models, some animal models, and also some studies involving patients where we're starting to have a look at these vesicles, so these little bubbles that are getting released by the cell in these really early disease mechanisms. So we, excuse me, So we grow cells in culture. So you can actually grow cells on a little plastic, basically like a little plastic, it's a flask. You can grow the cells on the flask, and you can have a look at the vesicles that get released. And you can model, I suppose, some of the processes that go on in early Parkinson's disease. So we've been doing that in the laboratory and then isolating these vesicles. And we have a look at all the molecules that are inside those vesicles. And the reason we do that is so that when we start having a look at patient fluids, so patients' blood, some nasal fluids, we couldn't try and identify some of those same molecules with the idea of going, okay, well, what we ideally want to do is be able to capture someone that's potentially going to develop Parkinson's disease, but capture them much early before they've got these sort of debilitating motor impairments.
Yeah, no, I can see the value, being able to identify early and then potentially be able to treat early and make decisions early for the person with Parkinson's. Fantastic. So we often ask people, what are the next steps with your disease or your research into Parkinson's? Like, so we've sort of started to identify the bubbles and the molecules in the bubbles. What are the next plans for you in the research?
Yeah, so for us, this is, I suppose, the research is at a really early stage. And as I said, some of these discussions sort of come out of coffee over a year ago, and we're just sort of starting to really understand the composition of these vesicles in early disease. And so the next stages are just to continue on evaluating these vesicles, but in some animals and also in some patients. And we've been really fortunate with there's two researchers at the Florey, Kevin Barnum and Leah Beecham, who have started up a trial whereby they are collecting bloods from patients and also some nasal fluids. So we're going to be, I suppose, translating what we've found in the animal models and in the cell models to patients. And so we will be isolating those vesicles out of patient fluids and seeing if we can detect, I suppose, that same signature that we see in the models in the laboratory in patients. So yeah, this is all a new area for us, but we really see it as important and I suppose under-researched. The vesicle field at the moment is really, I started in this area a little while ago, and not a lot of people had heard of these bubbles, but now a lot of researchers and industry and research development companies are realizing the value of really, I suppose, being able to look inside the brain without actually having to look inside the brain. So you're looking at a little bubble instead.
Oh, great. So a lot of people with Parkinson's are very interested in ongoing research. The research that you have at the moment, is that still recruiting patients or is that fairly closed off at this moment?
No, it's not closed off. So unfortunately, COVID has made the situation extremely difficult. So it's paused at the moment. However, if people are interested, they can contact our communications team at the Florey, and we can put them in contact with the Movement Disorders Clinic. And we can take it from there. But I think COVID has
it's very hard to do that.
Yes, it's difficult at the moment. But we're lucky that our laboratory, we can still undertake still continue the work on at the moment in regards to the samples that are in the freezer, and also our cell and animal work as well. So we're very fortunate in that respect.
No, that's great. Okay, well, this has been a fascinating interview because I knew nothing about vesicles or bubbles beforehand. So thank you so much for your time today, Laura. We'll get those contact details and we'll put them on the web with this video so that if there is anyone interested in participating or finding out more about Laura's research, they can find out how to contact you. And I think that's it. Anything else you want to impart?
No.
More wisdom.
Thanks for your time, Jodette. Much appreciated.
No, it's very interesting. No, thank you very much. And best of luck with your research. It sounds really interesting.